Translation refers to the process of polymerization of amino acids to form a polypeptide. The order and sequence of amino acids are defined by the sequence of bases in the mRNA. The amino acids are joined by a bond which is known as a peptide bond. Formation of a peptide bond requires energy. Therefore, in the first phase itself, amino acids are activated in the presence of ATP and linked to their cognate tRNA, a process commonly called as charging of tRNA and or amino acylation of tRNA to be more specific. So, if two such charged tRNAs are brought close enough, the formation of peptide bond between them would be favored energetically. The presence of a catalyst would enhance the rate of peptide bond formation. The cellular factory responsible for synthesizing proteins is the ribosome. The ribosome consists of structural RNAs and about 80 different proteins. In its inactive state, it exists as two subunits, a large subunit and a small subunit. When the small subunit encounters an mRNA, the process of translation of the mRNA to protein begins. There are two sites in the large subunit for subsequent amino acids to bind to and thus be close enough to each other for the formation of a peptide bond. So the ribosome also acts as a catalyst, means 23S rRNA in bacteria is the enzyme ribozyme for the formation of peptide bond. A translational unit in mRNA is the sequence of RNA that is flanked by the start codon AUG and the stop codon and codes for a polypeptide. An mRNA also has some additional sequences that are not translated and are referred to as untranslated regions, means UTR. So the UTRs are present at both 5' end before start codon and the 3' end, means after stop codon. They are required for efficient translation process. For initiation, ribosome binds to the mRNA at the start codon that is recognized only by the initiator tRNA. The ribosome proceeds to the elongation phase of the protein synthesis. During this stage, complexes composed of an amino acid linked to the tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with the tRNA anticodon. So the ribosome moves from codon to codon along the mRNA. Amino acids are added one by one, translated into polypeptide sequences dictated by DNA and represented by mRNA. So at the end, a release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. Now, regulation of gene expression. Regulation of gene expression refers to a very broad term that may occur at various levels. Considering that gene expression results in the formation of a polypeptide, it can be regulated at several levels. In eukaryotes, the regulation could be exerted at transcription level, processing level, transportation of mRNA from nucleus to the cytoplasm, and translational level. So the genes in a cell are expressed to perform a particular function or a set of functions. For example, if an enzyme called beta galactosidase is synthesized by E. coli, it is used to catalyze the hydrolysis of disaccharide lactose into galactose and glucose. So the bacteria use them as a source of energy. Hence, if the bacteria do not have lactose around them to be utilized for energy source, they would no longer require the synthesis of the enzyme beta galactosidase. Therefore, in simple terms, it is the metabolic, physiological, or environmental conditions that regulate the expression of genes. The development and differentiation of embryo into adult organisms are also a rest of the coordinated regulation of the of expression of several set of genes. So in prokaryotes, control the rate of transcriptional initiation is the predominant site for control of gene expression. In a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory proteins which affect its ability to recognize start sites. So these regulatory proteins can act both positively, means like activators, and negatively, means repressors. So the accessibility of promoter regions of prokaryotic DNA is in many cases regulated by the interaction of proteins with sequences termed as operators. So the operator region is adjacent to the promoter elements in most operons, and in most cases the sequences of the operator bind a repressor protein. Each operon has its specific operator and specific repressor. For example, lac operator is present only in the lac operon and it interacts specifically with lac repressor only. Let's read lac operon. So the elucidation of the lac operon was also a result of close association between a geneticist Francis Jacob and biochemist Jacques Monod. They were the first to elucidate a transcriptionally regulated system. In lac operon, where lac refers to lactose, a polycistronic structural gene is regulated by a common promoter and regulatory genes. Such arrangement is very common in bacteria and is referred to as operon. So, to name few such examples like lac operon, TRP operon, ARA operon, his operon, wall operon, etc. So the lac operon consists of one regulatory gene, the I gene. Here the term I does not refer to inducer, rather it is derived from the word inhibitor. And three structural genes like Z, Y, A. The I gene codes for the repressor of the lac operon. Z gene codes for beta galactosidase, means beta gal, which is primarily responsible for the hydrolysis of the disaccharide lactose into monomeric units galactose and glucose. The Y gene codes for permease, which increases the permeability of the cell to beta galactosides. The A gene encodes a trans-acetylase. Hence, all the three genes products in lac operon are required for metabolism of glucose. In most of other operons as well, the genes present in the operon are needed together to function in the same or related metabolic pathway. So lactose is the substrate for the enzyme beta galactosidase and it regulates the switching on and off of the operon. Hence it is termed as inducer. In the absence of a preferred carbon source such as glucose, if lactose is provided in the growth medium of the bacteria, the lactose is transported into the cell through the action of permease. Remember, a very low level of expression of lac operon has to be present in the cell and all the time. Otherwise, lactose cannot enter the cell. So the lactose then induces the operon in the following manner. The repressor of the operon is synthesized all the time constitutively from the I gene. The repressor protein binds to the operator region of the operon and prevents RNA polymerase from transcribing the operon. In the presence of an inducer such as lactose or allolactose, the repressor is inactivated by interaction with the inducer. This allows RNA polymerase access to the promoter and Transcription proceeds. Essentially, regulation of a lac operon can also be visualized as a regulation of enzyme synthesis by its substrate. So remember that glucose or galactose cannot act as inducers for lac operon. Can you think of for how long the lac operon would be expressed in the presence of lactose? Regulation of lac operon by repressor is referred to as a negative regulation, and lac operon is under control of positive regulation as well. But it is beyond the scope of discussion at this level.